we are having, if strong oxidizer gets into the bloodstream and methemoglobin is formed. It is a compound where iron 2 becomes iron 3. What has to be done to save the patient? You know that when your hemoglobin is not binding with oxygen, you are going to have a serious problem. You are going to have a serious problem. So what do? You change the blood, or you do what? Transfusion. You do your transfusion. So here you do what? You do interchangeable hemotransfusion has to be done. Transfusion has to be done. Transfusion has to be done. Okay. A 10-year-old child, guys, okay, let me drink this water. Thank you, guys. Sorry. A 10-year-old child complains of weakness, nausea, irritability, helmets of white color, 5 to 10 centimeters long, were found on the underwear. Guys, if you see something like perianal folds under the underwear, white color, microorganism, please and please again, don't think twice about your answer. Simply go in for what? Enterobins vermicularis. Enterobins vermicularis. Please, please. So again, we have what? Scraped from the perianal fold. That means under the anus. There is achromic oval of unsymmetrical form were detected. Please and please again, your answer is what? Is A. Vermicularis, vermicularis, enterobins vermicularis. Please, please. And you'll be seeing them often. Patient's parent with ill child came to the infectionist. They worked in one of the Asian countries for a long time. Child has earthy colored skin, loss of appetite, laxity, enlarged liver, spleen. What protozoa is this child likely to have? What protozoa is this child likely to have? First of all, they're giving you a country history or travel history where they work in what? In ancient countries. So over here, if you go to ancient countries, what is common is what? Leishmaniasis. So you're looking at what? For visceral leishmaniasis. And leishmaniasis is a protozoa that is caused by what? A leishmania. It is also called the black fever. And it is spread by a bite of sand flies. Again, it is also called the black fever, and it is transferred by a bite from the sand flies. And this disease or leishmania has 3 ways or 3 processes. We have what we call the cutaneous, the mucocutaneous, and then the visceral leishmaniasis. Cutaneous, mucocutaneous, and then visceral. So over here, it's affecting the internal organs, and then some internal organs plus the GIT. So we're talking about what? The visceral. Visceral means internal. Visceral. So here, you are thinking of what? Visceral leishmaniasis. Okay. White head, blue eyes, born in a healthy parent. Irritability, anxiety, trouble sleeping, feeding developed in the first month of life of the infant. What method of genetic investigation should be used for diagnosis? Look, these are what? Features, okay? These are features. These are not gene thing. These are not genes. So you don't want to say, let's do a genealogical like checking the history of the parents, line and those things. No, no. So over here, you just do what? A biochemical analysis. You do what? A biochemical analysis. Biochemical analysis. So again, we have what to call this cytological or cytogenetic. This detects abnormalities in chromosome structure. So if you're dealing with cytological, then we are talking about what? Chromosome structure or abnormalities in chromosome what? Structures. Now, biochemical deals with what? With protein functions. Protein functions and anxiety, trouble sleep, feeding abnormalities, white head, blue eyes are all what? Protein functionalities. They are all protein functionalities or enzyme metabolism. Enzyme metabolism. Enzyme metabolism. So basically, these are the things that we think of when we are talking about biochemical. Biochemical. So this is what E, biochemical. Why? Because it deals with what? Proteins. All of these are all proteins. White hair, blue eyes, feeding abnormality, trouble sleep. They are all enzyme metabolisms. Enzyme metabolisms. All right. We are having calcium channels of cardiomyocyte have been blocked. It has been blocked. What changes will happen? Don't forget, calcium are needed for what? Contraction. Calcium are needed for what? For muscle contraction or for cardiomyocyte what? Contractions. So if these calcium are not there, what it means is that the heart cannot contract properly. The heart cannot contract properly. So therefore, there will be decrease in rate and force of heartbeat. Decrease in rate and force, decrease in rate and in the force of what? Heartbeat. Yes, this is part, but of decrease force of contraction, yes, but also what? Heartbeat will also be decreased because if contraction is decreased, heart rate will also what? Decrease. Heart rate would also what? Decrease. And actually, this is also correct. This is also correct. So definitely decrease the rate and then the force of heartbeats. So therefore, your answer is E. Is E. After the trauma, the patient's vagus nerve was damaged. Again, when the vagus nerve or when the vagus nerve is damaged, what it means is that everything slows down. Everything, your heart will slow down because parasympathetic is involved, right? Parasympathetic is what is involved. Parasympathetic is involved. So if this nerve is damaged, everything will go high, not down. It will go high. Everything will go high. Everything will go high. Please, sorry. Because the vagus nerve is a parasympathetic nervous system which slows the heartbeat. So if it is irritated a little bit, what happens is that it will lead to a decrease in what? Heart rate or heartbeat. But over here, they say the vagus nerve is damaged, whether it is cut or whatever. But definitely there will be what? Evaluation of the automatism of the heart. Automatism of the heart, we are referring to what? We are referring to the SA node, the AV node, the bundle of His, the Purkinje what? Fibers. SA node, the atrioventricular node, the bundle of His, and the Purkinje what? Fibers. But of course, from the atrium level, we are dealing with what? The SA node. We are dealing with what? The SA node. So this is also referred to what? The case flex node. So there will be evaluation of automatism of a case flex node. A case flex node. So this is what we are what referring to, or we will see evaluation in this node or SA node when there's what? A damage to the vagus nerve. A damage to the vagus nerve. So if it is irritated a bit, it can lead to what? Decrease. If it's completely cut away, then it can lead to what? Increase. Okay. So valuation will be there. So A should be your answer. In the ovary specimen colored with hematoxylin Eosin follicle is determined where cubic-shaped follicle epithelium cells are placed in a 1 to 2 layers. Just 1 to 2 layers. And a scarlet covering is seen around the oocyte. What is this follicle? Please write this thing down. Write this thing down. Features of all of these things, or some of these things. For primordial features, right? Primordial. What do you see under primordial? You will see a dominant, small, only one layer of flat granulosa cells. Again, only one layer of a flat granulosa cells are seen. It is dominant but small, and only one layer. So that is what primordial features. I'm saying this because if you get to go to exams and you see only one layer, dominant cell, you are thinking of what a primordial. What about primary? Primary include mitotic cells. Then we have cuboidal granulosa cells, usually 1 to 2 layers. I repeat, it is usually 1 to 2 layers. So in primordial, 1 layer. In primary, 1 to 2 layers. 1 to 2 layers. Now in secondary, we have what we call theca cells. Theca. Theca cells. Then we have multiple layers of granulosa cells. Multiple layers. Then, last but not the least, we have the mature or the tertiary features. Mature or tertiary follicle. Mature or tertiary. It includes granulosa and theca cells, or increase in granulosa and theca cells. They become more than the secondary. And sometimes you will see what we call an antrum volume. Antrum, A-N-T-U-M. T-R-U-M, antrum, A-N-T-R-U-M volume. Sometimes you will see an increase in the antrum volume in a mature or tertiary follicle. So again, over here, we are talking about 1 to 2 layers. So what comes to mind? Primary. So the answer is A. While studying of a family tree with a history of hypertrichosis, that's hyperhirsutism of the ear, that when there's too much hair in the ear. This sign was found only in men. Guys, look, it was found only in men, and it was inherited from the father to the son. Again, a father is always what? XY. So what chromosome would a father donate to the son? Of course, Y. The father will always donate Y to the son. What does it mean? What it means is that this problem is related to what? To the Y chromosome. So this is connected to Y chromosome. So your answer is A, Y chromosome. Again, if they tell you it is connected to only men, you know the chromosome number for men, or the sex chromosome for man is XY. And you know that man will always give X to the female children, and will give Y to the male children. So if this disease was inherited from the father, that means it was from the Y. Again, guys, look at this. Same question. Perianal folds of a 5-year-old girl. White worms. You remember? White. The worms were sent to lab. Physicians saw what? A filiform helmet. Guys, what is your answer? I've told you guys that anytime you see this, you are looking at what? Enterobins vermicularis. And the disease is what? Enterobiasis. So your answer is what? Is D. enterobiasis, enterobiasis, enterobiasis. Now, these two, just a revision, I mentioned earlier on, they are caused by what? Salted fish or undercooked salt fish. Undercooked salt fish. That's just by the way. So let's continue. Part of the DNA chain turn about 180 degrees due to gamma radiation. What type of mutation is it? Guys, look, listen, this is the head. This is the tail. Okay. And I think that this chromosome has been turned 180 degrees. So turn 180 degrees means that this will now be the head. This will now be the what? The tail. So this 180 degrees turn. That means that this chromosome has been what? Inverted. So this is called inversion. It is called inversion, inversion, inversion. Now, when we say replication, it means what? Duplication. When we say deletion, it means part of the chromosome has been deleted. And when we say translocation, it means part of this has been removed to go and complete the other chromosome, and the other chromosome has been removed to come and complete this chromosome. That's called translocation. It will move from one chromosome to the another. It will move from one chromosome to the other. So that is what we call what? So this is what? Inversion. Because 180 degrees. Inversion. I love this. 46 chromosomes were revealed in a karyotype. Now, on the 15th pair of the chromosome, they say one of the 15th pair of the chromosome is longer than usual due to connected chromosome from 21 pair. What it means is that chromosome has moved from the 21 to join the 15th pair or one of the 15th pair. And that phenomenon is called what? Movement from one chromosome to another one is called what? Of course, translocation. Translocation. I've said this. Deletion means what? It has been deleted and thrown away. Inversion means what? It has turned upside down or 180 degrees. Duplication also means what? Replication. That means it has doubled. So please take note of these terminologies and you will not have any problem. So your answer is D. Again, look at this. Pregnant woman has been having toxicosis with severe repeated vomiting. Repeated vomiting. The question is, what acid-base imbalance will you see? Forget about everything. There's repeated vomiting. Acid-base. I've said this one countless number of times. What do you feel in your nose? Sorry, in your mouth? You feel sadness or bitterness. And that bitterness is what? It's acid. That means acid are being secreted when you vomit. And so therefore, if acid are secreted, what will be left inside you will be what? Will be alkaline. And so therefore, we're having what? Excretory alkalosis. Excretory alkalosis, also known as non-gaseous alkalosis or metabolic alkalosis. All of these things mean the same. I have said this one like a billion times. So please, if you've not watched the video, do justice to the videos, and all of these will make sense to you. So your answer is C. Great. You remember when we talk about the immature messenger RNA, right? Great. Look at it. The general structure of an eukaryotic gene is as follows: exon, intron, and exon. You remember when I mentioned about the exon, intron, and exon, right? That they form part of what? An immature DNA, sorry, an immature RNA. Now, such functional structure of a gene leads to certain specifics of the transcription process. What sequence corresponds to the precursor of the messenger RNA? And again, look, anytime they ask you this question, please maintain what was in the question. I repeat, they have what? Exon, intron, exon. Exon, intron, exon. So the sequence will also be what? Exon, intron, exon. Exon, intron, exon. So again, you are looking for what? For A as your answer. A. A is your answer. A is your answer. All right. But however, when it's going to be, or when it undergoing transcription or translation, they'll be what? Some of them will be removed. Some of them will be what? Spliced. That's called what? The splicing. That is called the splicing. The splicing. So again, what you see the sequence, the same sequence you will see in your precursor messenger RNA. All right. Awesome. I mentioned some karyotypes to us the last time, or some abnormalities. We talk about Klinefelter, Turner syndrome, trisomy X, trisomy 21, or Down syndrome, Fatao syndrome, Edwards syndrome. We talk about all of these things. However, let's look at this question. Medical examination at the military registration and enlistment office revealed a 15-year-old boy. So first of all, this person is a boy with inadequate body proportions. Gynecomastia means sort of breast anyways. And this is a female thing. And look, a female pattern of pubic hair distribution. Female pattern of pubic hair distribution. What it means is that this boy is having some signs and features of a female. The boy also had fat deposit on the thigh, no facial hair, high voice, subnormal intelligence. What karyotype is this corresponding to? Again, we are having a boy with female characteristics. So that means that this person is called what? A Klinefelter. Klinefelter. And so therefore, we're going for what? XXY. A male with a female features. Male with a female features. Also known as Klinefelter's syndrome. Klinefelter's syndrome or Klinefelter disease. Klinefelter. 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 So your answer is A. Is A. RNA that contains AIDS virus penetrated into a leukocyte, and by means of reverse transcriptase, guys, look at this. Reverse transcriptase force a cell to synthesize a viral DNA. This process is based upon what? Guys, they are telling this what? A reverse transcription. Reverse transcriptase. That means it controls reverse transcription. Reverse transcription. So this one, you are thinking of what? Reverse transcription, because we are using reverse transcriptase. Reverse transcriptase. So again, you are looking for what? For a reverse transcription. A reverse transcription. Reverse transcription. So that means that you are synthesizing what? A DNA on a messenger RNA. Okay? You are trying to synthesize DNA on the matrix of a viral messenger RNA. You want to represent DNA from RNA. Aha. Good. According to the model of a double DNA helix that was suggested by Watson and Krok, it was established that one of the chains will not be lost during replication. Again, one of it will not be lost during replication, and the second chain will be synthesized complementary to the first one. What way of replication is it? What way of replication is it? What way of replication is it? So first of all, let's look at the different types of replication. I'll go with the two main. That is what? Semi-conservative and then conservative. Semi-conservative and then what? Conservative. When we say conservative way, or by this mechanism, one daughter cell receives original DNA molecule, whilst the other one receives a completely different new copy. Again, in a conservative replication, one daughter cells receives the original DNA molecule, whilst the other one receives completely different new copy. New copy. So what it means is that, assuming you have this, okay, and out of this, you're going to have a replication. You are to have two daughters. That means double. So you have 1, 1. And this one is identical to this one, but this one is non-identical. This gene is completely new, or this copy is completely new, but this is like original from the previous one or the mother one. So that is what conservative. Now, in semi-conservative, the original DNA is split into two strands, and each strand acts as a template on which a new strand is synthesized. That means that if you have this, it divides into what? Into this and that, upon which a copy is made from each other. So that means this one are taken, or they are all identical to the parent one. To the parent one. But look at this. According to the model of a double DNA helix, it was established that one chain, one chain will not be lost during replication. That means one chain would be what? Identical to the parent one. The second chain, that means one chain will be different, which is this one. Because one chain will not be lost during replication, and the second chain would be synthesized complementary to the first one. Complementary to the first one. So that means this one is identical, one is not identical. One is identical, one is not identical. If I'm correct in this scenario. And so therefore, we are referring to what? A semi-conservative. Semi-conservative. Wait, what am I saying? They said one chain will not be lost during replication, and the second chain will be synthesized complementary to the first one. Guys, you're referring to this one. That means that no thing will be lost. That means the daughter cells will be identical to the parent one, or they will have features in the parent one. So actually, this one is what? Semi-conservative, not conservative. This is semi-conservative, not conservative. Semi. That means they complement the parent one. Because look at it. The second one will not be lost during the replication, and the second one is complementary to the first one. That means this one is complementary to this one. This is complementary to that. This is complementary to that. And they're saying that this one, or this particular one, was not lost from this. That means this is the same as that. So please, this is English. Let's try to understand it. It's English. So here, your answer is what? Semi-conservative, not conservative. Semi-conservative. All right, so your answer is A. A teenager was irradiated with a high radiation dose that resulted in a serious changes of the lymphoid system. Lysis of many lymphocytes. Again, we said that when radiation is or comes upon or is being exposed, when is exposed to radiation, the cells that are normally lost, first of all, are what? The lymphocyte, or they are affected the most. The lymphocyte. Now, restoration of a normal hemogram is possible due to the following gland. The question now is that which of these glands produces lymphocytes, or is lymphocyte produced from? Yes. And basically, you are thinking of what? Of the thymus. The thymus. The thymus. The thymus. So basically, immature cells are produced from the bone marrow, and they enter into the thymus for them to be matured. Again, the bone marrow produces all cell types, especially lymphocytes. The immature lymphocyte will enter into the thymus for maturation. For maturation, and of course, for storage. Of course, for storage. So the answer is A. Good. Guys, the same question, but formulated in a different way. It was revealed that T lymphocytes were affected by HIV. Virus reverse transcriptase was used to catalyze the synthesis of what? Again, I told you guys that over here, it means that you want to what? Produce DNA from messenger RNA. You want to, because it's a reverse. It's a reverse. First of all, normal transcription is to produce what? Messenger RNA from DNA. So usually DNA to RNA. to RNA, you use what? Transcription. DNA to RNA. Now from RNA, the opposite direction, from RNA to DNA, you use the reverse transcription. That means you are trying to produce what? DNA from RNA. DNA from RNA. So over here, you are looking at what? For DNA on the matrix of messenger RNA. On the matrix of messenger RNA or virus messenger RNA. So here, your answer is D. Is D. So never get confused about this question because it's easy. It's easy. So that means this is what? A reverse transcription. So the other one, they were asking us, what was it? It was a reverse transcription. But what actually is going on is that DNA is being synthesized from messenger RNA. As simple as that. A patient, after a pathological process, has a thickened alveolar membrane. The direct consequences of the process will be the reduction of, look at it, there's a thick alveolar membrane, very thick alveolar membrane. And you know that this alveolar membrane is responsible for diffusion of gases. You know it is for diffusion of gases. That's the membrane in which oxygen will enter and carbon dioxide will go, or carbon dioxide will go and oxygen will enter. That is the membrane. So what happens? It means that there will be what? Diffuse lung capacity. Or what will be affected is that there will be a reduction of what? Diffuse lung capacity, or diffusion will be what? Affected. Diffusion will be affected. And you know that diffusion of gases depends on some 4 factors. By the way, diffusion of, in fact, any substance on a membrane is dependent on 4 factors. We have 1, the concentration gradient, 2, the gas solubility, 3, the membrane thickness, and then 4, the surface area. I repeat, the 4 factors by which diffusion of gases take place include what? The gradient or concentration gradient of the gases, the gas solubility, membrane thickness, and then the surface area of the membrane. Surface area of the membrane. So basically, that is it. So this is D. Over here, membrane is affected. So that means it will be difficult for gas to diffuse through. Autopsy of a newborn reveal polydactylia, microcephalia, chelocysis, uranocysis, whatever, as well as hypertrophy of parenchymatous organs. Look at this. They said this patient is having what? Patau. I've mentioned this and I'm mentioning it again. In Patau, we have what? Trisomy 13. In Edwards, we have trisomy 18. In Down syndrome, we have trisomy what? 21. Trisomy what? 21. So again, we are doing what? Patau. So for Patau, it's what? Trisomy 13. Trisomy 13. In Edward, trisomy 18. In Down syndrome, trisomy 21. Please take note. Guys, look at this again. I won't continue. Look at this again. Name of the follicle, 1 to 2 layer of cells. And your answer is, of course, primary. Please, let's not dwell on this topic. It's primary. I believe you are written them down. Okay, so let's continue. A patient has symptoms of inflammation of urogenital tract. That means the urinary tract, urogenital. Examination of the vaginal smear revealed monocellular paired shape, a pear shape, pointed spike at the posterior end, big nucleus, and undulating membrane. What protozoa is being described here? And this is what? Trichomonas vaginalis. Trichomonas vaginalis. And it is one of the major causes of vaginitis in women. One of the major causes of vaginitis in women. In women. In women. So please take note of that. And again, their shape is like a pear or pear-shaped. It's like a pear-shaped. And it has 4 anterior flagella. So add this onto it. It has 4 anterior flagella. It has a central nucleus, and a central nucleus is big, and then 4 anterior flagella. 4 anterior flagella. And the membrane is undulating membrane. So please write it down. So this is what? Trichomonas vaginalis. So your answer is E. Guys, again, look at this question. During examination for school children, look at the asymmetric oval X with a larva in the scrape from her perianal folds. Perianal folds. That is your clue. So perianal folds, you are thinking of what? Of course, enterobiasis. Or enterobens vermicularis. The same thing. A woman delivered a dead child with multiple developmental defects. What protozoa might have caused the intrauterine death? Intrauterine death. So over here, you are looking at what? A touch infection. Touch infection. So when you are pregnant and you give birth to a dead baby and there's an infection, you are looking for what? A touch infection. That is TORCH. Where T stands for toxoplasmosis. That is TO, toxoplasmosis. The R stands for rubella. C stands for cytomegalovirus. H stands for herpes simplex. Herpes. Again, toxoplasmosis, rubella, cytomegalovirus, and then herpes simplex. So over here, which one of them is a touch infection? Of course, toxoplasmosis. Toxoplasmosis might have been the reason why this woman delivered a dead baby. So your answer is B. Okay. Look, tuberculosis is detected by means of combined chemotherapy that includes substances with different. Question is, what anti-tuberculous medication inhibits transcription of RNA into DNA? It inhibits transcription of RNA into DNA, which, of course, all of these are used for treatment. Apart from this one, all of them, the rest are used for treatment of, tuberculosis. But which one of them has the function of inhibiting what? Transcription. And your answer is what? Rifampicin. So write this down. Rifampicin blocks transcription. It blocks transcription. That is RNA synthesis from DNA. It blocks transcription. Good. Isoniazid. They inhibit the synthesis of mycolic acid. Mycolic acid. MYCOLIC. Mycolic acid. And these will destroy the cell wall. Ethionamide. This has a structural analog like the isoniazid. So it's like the isoniazid. So it means that it also what will inhibit the synthesis of what? A mucolic acid or mucolic acids. It has the structure of what? Isoniazid. Whilst the para-aminosalicylic acid, also called the PABA, para-aminosalicylic acid, also called the PABA. This is also a competitive inhibitor for PABA in folate synthesis. In folate synthesis. In folate synthesis. And again, folic acid, as you know, are used to undergo what? Differentiation of cells. Differentiation of cells. So if this is blocked, that means the cells will not be what? Differentiated, and it means the cell will not grow. So basically, I believe you have written them down, so that will help you to differentiate between the mechanism of actions or what they really do. Okay, what they really do. So again, rifampicin prevents what? Transcription. Isoniazid prevents what? Mucolytic acid, the same as itonazide. And then the PABA acid also is involved in what? Folic acid. So it's competitive inhibitor of PABA in folic acid. As simple as that. So your answer is what? It's rifampicin. Rifampicin. All right. A patient experienced a sudden temperature rise, 39, 6 hours, temperature was normal. Second day, it reoccurred. There is what? Paroxysms. There is, in the period of what? Paroxysms, the temperature reached 41. Again, a pyrexia period began after 8 hours. What type of temperature is it? This is seen in people with malaria. So those those are Africans, or those who have been to Ghana. This is typical for malaria. This is typical for malaria. And this kind of temperature where it goes and comes, we call it what? Intermittent fever. We call it intermittent fever, where there's alternation of fever, where it goes and comes, alternate with the normal temperature, temperature. So that is common in what? In malaria. So what about the recurrent? In recurrent fever, we have rising temperature and normal level of what? Temperature. But the rising temperature, the number of days is more. For example, in rising temperature, you can have about 5 to 8 days. 5 to 8 days. Then it will come back to normal. Then again, it will increase again. And again, these kind of temperature are found in recurrent typhus. And sometimes, not common, but sometimes too, you can see it in malaria patients. So that is what recurrent. But this one, it is just the same day, it keeps changing. That's why it is what? Intermittent. In febris hectica, or in hectic fevers, it is what we call exhausting fever. Then it will make you very weak. It will make you what? Very weak. That's what? Exhausting fever. And this is very, very common in sepsis, tuberculosis, and so on. So as people with sepsis, they get tired easily because the fever is just hectic. Hence the name hectic fever. So basically, these are the main ones you can be thinking about. We also have febris continua or persistent fever. We have remittent fever or remittent fevers. We have all of those things available. But basically, here is what? Intermittent or intermittent. All right. Larvae were detected occasionally on a microscopic examination of a patient with pneumonia. Look at it. Eosinophils were detected. So if eosinophils were detected, you are thinking of what? Ascarias or ascariasis. We call it Ascaris lumbricoides. Or ascariasis. That's the disease it causes. Again, you are looking at what? Eosinophils. So please write it down. Eosinophils. All right. Okay. You are studying functioning of a bacterial operon. The operator gene has been released from the repressor gene. Immediately after the following process will start in the... Actually, the operon is actually used for what? Transcription, basically. It is used in transcription. So that means when you start the operator gene, what it means is that you are starting with what? With transcription. You are starting transcription. So please just take note of that. You are starting with a transcription. A transcription. All right. Now again, when the repressor gene is stopped, what it means is that transcription will also stop. So what you're doing with transcription. All right. Like this question. While studying maximally spiralized chromosomes of a human karyotype, the process of cell division was stopped in the following phase. So usually, when chromosomes are aligned at the equator, that's over here, that is where cell division can stop. Or that is where you can give a medicine to stop what? Cell division. And that phase is called what? The metaphase stage. The metaphase stage. The metaphase stage. So this is the cell division. This is the place whereby cell division can be stopped. Cell division can be stopped. It is inside the equatorial plane. The equatorial plane. So your answer is metaphase. Metaphase. So D is your answer. D is your answer. It is known that the gene responsible for development of blood groups according to the ABO system has 3 alleles. If a man has blood group 4 means what? AB. Am I right? Yes. It can be explained by the following what? Variability form. So if someone is having what? A blood group AB, what it means is that he is having a combination or a combinative form of variability. Combinative form of variability. Why? Because it involves 2 alleles of genes. 2 alleles of genes. That means is from blood group A and blood group B is giving us what? The blood group AB. Blood group A, blood group B gives us what? The AB. So example, if you are AO, that means you are A, and your father is OB, that means it's what? It's B. So the only way they can give you AB is because this is coming from here. So we give it AB. 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 So if you're a child with blood group AB, go and check your parents. Either they have, one is having OA, or one is having what? The AB. If one is having the AB, that means the other one, the house, might be having what? O or anything. That's just by the way. No, the person cannot have O. The person cannot be O. 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 So if you go and your mother is O and your father is AB, you have to do a paternity test because that might not be your dad. That was just by the way. So your answer is what? Combinative. 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 All right. Again, we are done with part 2, and the last part is not actually much. It's not actually much. So someone will say, let's close our eyes and finish it. So let's close our eyes and finish it. Then you ask your question, okay? Let's discard. Where is this? The last one. Good. Please let me hear from you guys. Are you guys sleeping? Let me hear from you guys. Yima, can you hear me?
Yes, sir. Break.
You said, let's go for break, eh?
Yes, I think I want to use the washroom.
Okay, then fine. Then 5 minutes break will do. But we'll not ask questions until we finish the presentation, okay? Okay. So guys, let's have the break. Let's have the break. So I'll pause the recording. Apparently, this is going to be our last tutorial on biology. So let's just move. Good. We have a patient complaining of pain in the area of his liver. Duodenal intubation revealed yellowish over narrowed at the pulse X with an operculum. Guys, operculum at the end. Size of these X is the smallest among all helminths. It is the smallest among all helminths. So, what is your most probable diagnosis? Again, look at the complaint. In the liver. So this is what? Liver fluke. Liver fluke. And I said another name for liver fluke is what? Opesco opistocosis. 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 That's another name for what? For liver fluke. And again, I said it can be obtained from what? From uncooked or undercooked salty fish. Undercooked salty fish. Undercooked salty fish. And again, it is the smallest among all elements. It is the smallest among all elements. And again, the operculum again should direct you that this is opistocosis. So your answer is C. Your answer is C. All right. A patient consulted a urologist about pain during urination. An analysis of his urine taken in the daytime revealed X with a characteristic sharp point. It is known from analysis that this patient has recently returned from Australia. What is the most likely diagnosis? Guys, even if you don't know what this disease is, since this is a urinary tract infection, or this has to do with urination, so definitely the answer is what? Urogenital schistosomiasis. Urogenital schistosomiasis. Again, even if you don't know anything at all, know that this has to do with what? With urine. So urogenital. With intestine, definitely I think with the intestine, right? Good. And with opistochiasis, you already know what it is. So over here, you're going for A. For A. All right. You're going for A. Again, look at this question. Child is complaining of a general weakness, loss of appetite, trouble sleeping, itching in the perianal area. Again, itching. So how will you diagnose this patient? Of course, scraping from the perianal folds. Scraping from perianal folds. Again, we have done this one. And again, look at it. This is what? Enterobiasis. Because of what? Perianal folds. Enterobiasis. We've discussed this like million times. So this question, please, is one time and you move. One time and you move. The cell of the lab animal was overdosed with Röntgen rays. As a result, albumin or albuminous fragment formed in the cytoplasm. What cell organelle will take part in their utilization? So now cells that have been overdosed with radiation rays means that the cells have been exposed to high doses of ionizing radiation. It means it has been exposed to what? High dose of ionizing radiation. And this radiation will cause what? Cellular degradation. It will cause cellular degradation due to the damage to the DNA. And again, what enzyme is involved in the destruction of cells? Yes, lysosomes. These are involved in the destruction of cells. So it will take part in destruction of all these things, thereby leading to what? A lot of fragment of the albumins. So your answer is what? Is E. Look at this again. In the course of practical training, students studied a stained blood smear of a mouse with bacteria phagocytosed by the leukocyte. What cell organelle complete digestion? Complete the digestion. That means destroys the bacteria. Of course, you are thinking of what? Lysosomes. Lysosomes. They are involved in degradation of cells or destruction of cells, destruction of bacteria. So again, lysosomes. Lysosomes. So your answer is E. Lysosomes. Lysosomes. All right. Okay. I love this question, and I want us to solve this question. They said a woman with blood group B, rhizos negative. Do you guys remember when I mentioned something about rhizos negative? That they are not common. They are rare. Majority of the people are what? Positives. But there is a woman with negatives. And again, I told you guys that with negatives, they normally will lead to what? A problem if they don't give them anti-rhizos medication. So look at it. This woman is B, gave birth to a child with blood group A. Now, the child is diagnosed with hemolytic disease of the newborn as a result of rhizos incompatibility. As a result of rhizos incompatibility. So what blood group is the child's father? So definitely the child father will definitely be rhizos positive. Be rhizos positive. Because for the child to have rhizos incompatibility, it means that there is the presence of rhizos factor in the father. In the father. So that means that, first of all, you have what? Rh positive. But look at it. Mother is B. Child is A. That means that this A did not come from the mother. It didn't come from the mother. It definitely came from the father. And so therefore, you are looking at this as blood group what? A. So I is blood group what? A. I is blood group what? A. So here, your answer is what? Is A. Your answer is A. Blood group A, rhizos positive. Sorry, this is II. This is II. Blood group I is O. This is what II. So this is what blood group A, rhizos positive will be the father's, blood group. 2 days after consumption of pork, smoked pork, that's pig, right? Patient got face and eye edemata, gastrointestinal disturbances, rise in temperature, muscle pain. There is eosinophilia. There is what eosinophilia. What element could this be? What element could this be? Now, if they have not brought this history there, I tell you that if you see eosinophils, what comes to mind? You are thinking about ascaris. You could be thinking about ascaris. You could be thinking about ascaris. But for the fact that they've brought this history in this patient, please, this is trichinosis or trachina. Trichinosis or trachina. And this is a roundworm. It is a roundworm. And they are found in undercooked meat, especially pork. They are found in undercooked meat, especially pork. So the history will make us go for what? Trachina. But in the absence of it, we could have been thinking of what? Ascariasis. All right. A lymph node punctate of a patient with suspected protozoal disease was examined. Examination of the stain specimen revealed crescent bodies with pointed end. Look at this: blue cytoplasm, red nucleus. Please, this is typical for toxoplasmosis or toxoplasms. Red nucleus, blue cytoplasm. Red nucleus, blue cytoplasm. Chelsea and Manchester. This is what? Toxoplasmosis. Chelsea, Manchester is toxoplasmosis. So in exams, if you forget anything at all, remember Chelsea and remember what? Manchester United. And that gives what? toxoplasmosis. So your answer is what? Is A. The guide of the scientific uh, expedition in India was native who always was with his dog. What invasive disease can be transmitted by the dog if it is the source of infection? If it is the source of infection. And basically, your answer is what? Echinococcus. Echinococcus. Echinococcus, yeah. And these echinococcosis are normally infected, or they infect animals such as dogs, foxes, and wolves. So dogs, foxes, and wolves. Foxes, wolves. Foxes and wolves. Foxes and wolves. Foxes and wolves. Echinococcus. Dogs, wolves, foxes. So the answer is D. A patient with acne on his face, microscopic examination of the scrapings from the affected area revealed living porate vermiform arthropoda with 4 pairs of short extremities in the front part of their bodies. What lab diagnosis are you looking at for? What lab diagnosis are you looking at for? This person has what? An acne on his face. Acne on his face. So it has to be with what? With the skin. With the what? With the skin. With the skin. So over here we are looking at what? Demodicosis. 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 And we know that pediculosis, we know of scabies, and so on and so forth. We'll come to myiasis in one of the questions. But that's what? Demicosis. Demicosis. And so sometimes we also call it the red munch. Red munch. Red munch. Red munch. MANGE. Red munch. Red munch. Red munch. Red munch. So, yeah, basically, that is just it. And yeah, it can affect, or they can live inside the hair follicles, all these places, the legs and all that. And by their characteristics, unfortunately, I didn't have a picture of it to show you how the disease is called demodex, how demodex looks like. How demodex look like. But it looks like what they've described over here. 4 pairs of legs. The larva, the nymph have only 3 pairs. The larva and nymphs have only 3 pairs. Only 3 pairs. So we're looking at what? At demodicosis or demodex infection. Demodex infection. A family of students who came to Africa got a child with anemia. The child died soon. Examination revealed that the child's erythrocytes have abnormal semilunar shapes, specify the genotypes of the child's parent. Look at it. People from Africa, the child's parent. So that means that it could be that the parents are actually normal, okay? They are only what? Carriers. They are what? Just carriers. And that's how they married and gave birth to a child with what? With sickle cell. With sickle cell. So being a carrier means A, small a. A, small a. As simple as that. So your answer is what? Is D. Is D. So and don't forget that sickle cell is a recessive gene. So this AA must be found on the chromosome before it can exhibit its function, or before it can show that it is a sickle cell. It is a sickle cell. So parents are carriers. So AA. Examination of a cell culture got from a patient with lysosomal pathology. So lysosomal pathology, you know it, revealed accumulation of lipids in the lysosome. What disease is it? Lysosomes in the lipids. Sorry, lipids inside the lysosome. We are referring to what we call the Tay-Sachs syndrome or Tay-Sachs disease. We've done this one, I think, in biochemistry. We have done this one in biochemistry. So this is what lysosomal diseases. Lysosomal diseases. And when the lipids are accumulated in the in the lysosomes, we call it what? Tay-Sachs disease. We call it Tay-Sachs disease. Tay-Sachs disease. Tay-Sachs disease. And this can damage a lot of what? Cells. So it's Tay-Sachs. 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 You know your guy Wilson? This is what? Copper. Right? This is what? Copper. Gout. This was uric acid. Or purines. You remember them? Good. Galactosemia, we're talking about galactose. So you know all of these things. Then this one is what? It's phenylalanine that is found, right? So here is C. A 39-year-old woman has made seen in the region of Mamela, a small ulcer with inflammatory hyperemia and cutaneous edema. Histologic examination of the tissue sampling from the area revealed malphagian layer of thickened epidermis. These are what? Atypical cells. Atypical cells with light and optically empty cytoplasm. No intracellular bridges. Such cells were also found in the orifice of the big mammal gland duct. What is your diagnosis? What is your diagnosis? Usually a large cell with a clear cytoplasm and eccentric are normally, which are normally found out on the epidermis. These are normally located, or they are called pungent cells. Cells which are large with a clear cytoplasm with even hyperchromic nuclei. That means the nucleus itself is hyperchromic. That means it is colored. In the epidermis, we call it pungent cell or pungent cells. Pungent cells or pageant cells. Pageant cells. So definitely, AIDS disease we call what? Pungent what? Disease. Pungent disease. Pungent disease. So this is pungent disease. Pungent disease. Again, you are looking out for what? For these empty cytoplasms. Empty cytoplasm with no intracellular what? Bridges. No intracellular bridges. No intracellular bridges. Okay. The Golgi apparatus exports substances from a cell due to the fusion of the membrane circle with the cell membrane. Again, Golgi complex exports substances from the cell due to the fusion of the membrane circle with the cell membrane. Now, the circle contains flow out. Flow out. What process exists? What process is it? Of course, this is talking about what? Exocytosis. Exocytosis, whereby the cell will begin to, how do I put it? It takes cell outside the cell. Okay? It takes substances. It takes substances outside the cell, or it empties it. We call it what? Exocytosis. Now, if it picks it up, we call it what? Endocytosis. Endocytosis. So, in endocytosis, substances that are external to a cell are brought into the cell. That is in what? In endocytosis. Substances outside are brought into the cell. Now, in exocytosis, what happens? It means that substances are also transported from the cell into the outside, into the outside, into the outside. And that's what exocytosis exocytosis. And that's what they say, that substances from a cell, that means they are inside the cell. Now, this cell begin to fuse with the circle, with the membrane. So, assuming that this is a cell, this is a cell, and this is a cell, and this is a substance that you want to undergo what? Exocytosis. This is a cell. And inside it, we have what? Granules or substances inside. So this is your bigger cell, and this is the substance you want to what? Excrete out or take it out. So what it means is that the cell membrane of this circle or this vesicle will begin to fuse with the cell membrane of the cell. They will fuse together. And when they fuse together, what happens? This part begins, so same cell. So, okay, sorry. Let me do it better. Aha. Now we have this. When they fuse together, it opens. And when it opens, the content begins what? To come out. And that's called exocytosis. Exocytosis. So your answer is what? Is A. Exocytosis. And that's what has been explained over here. The circle refers to what? A vesicle. Like this. This is a vesicle. This is a vesicle. Okay. So your answer is A. A patient working at a pig farm. Again, pig farm. Complains about paroxysmal abdominal pain, that is sudden abdominal pains, liquid feces with admixtures of mucus and blood. There's headache, weakness, fever. Examination of the large intestine revealed ulcers from 1 millimeter to blah, 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 blah. Feces contain oval unicellular organisms with cilia. Oval unicellular organisms with cilia. Unicellular organisms with cilia. So, over here, these are actually referring to what we call balantidiasis. Balantidiasis. Up until now, you might have heard about amoeba, protozoa, amoeba. You know of toxoplasmosis, lamblia, and so on and so forth. But this is referring to what? A balantidiasis, because it's a unicellular organism. It is a unicellular organism. Unicellular organism. And how do you diagnose this? Diagnosis is made by finding a large ciliated trophozoid or large cyst with a vein-shaped nucleus in the stool. That is how to know this microorganism. That is how to know microorganism. And it is very, very similar to Entamoeba histolytica, because it will end up boring holes in the colon, causing ulcer formation. That's what here, your answer is what? Balantidiasis. All right. A boy found a spider with the following morphological characteristics: 2 centimeters, large, roundish, black abdomen with 2 arrows of red spots. 2 rows of red spots on its dorsal side. 4 pairs of jointed limbs are covered with a small black hairs. What arthropod is it? What arthropod is it? Have we heard of what we call the European black widow or the window spider? Yes, this is the widow spider. The widow spider, also known as European black widow. European black widow. And again, this actually, if you go to Central Asia or Eastern Slavic regions, They call this black window or window spider, they call it Karaket spider or Karaquit spider. Karaket spider. Karaket spider. So it's the same thing. It's the same thing. And why do we call it a black window? Because it has what? Roundish black abdomen with 2 rows of red spots. So black abdomen, black window, or Karaket spider. So please take note, Karaket spider. So it's A. Normal actively dividing cells of the human red bone marrow are analyzed. What number of cells chromosomes are typical in G1 period? Sorry, guys. There's bone marrow division, and they're asking how many number of chromosomes are found in the G1 period. Now, the G1 phase is also called the gap 1 phase, and that is the first of 4 phases of cell cycle. It is the first of 4 phases of cell cycle. Now, in this part, actually, this are all taking place in the interphase. That is the resting stage of the cell, where the cell prepares itself for cell division. So during that stage, or during the G1 phase, there is what? Cell synthesis, or normally just in the interphase phase, there's what? Cell synthesis. There is cell synthesis, and then proteins in preparation for subsequent steps during or leading to what? Mitosis. For mitosis. And the G1 phase ends when the cell moves into the S phase. That is what? The synthesis phase of the interphase. And usually during the G1 phase, we have 46 number of chromosomes. I repeat, we have 46 number of chromosomes. 46 number of chromosomes. So please take note, 46 number of chromosomes. All right. A man has worked in an African country for 3 years. A month after he returned to Ukraine, he consulted an ophthalmologist and complained about eye ache, lipedema, lacrimation, and temporal visual impairment. Underneath the eye conjunctiva, the doctor revealed helmet of 30 to 50 millimeters with, guys, look, with long, elongated filiform bodies or filiform body. What is your diagnosis? From the name filiform, you have what? Filariasis. Okay, so this is what? A parasitic what? Disease. A parasitic disease. So your answer is what? Is D. Filariasis. So please look at it. Always look at it. All right. Hey, another question on cell cycle. Okay. There's another question on cell cycle. Okay. So cell cycle of a cell induces a process of DNA auto-duplication. Auto reduplication. As a result of this process, monochromatic chromosomes became bichromatic. This phenomenon is observed within the following period of cell division. So first of all, I didn't explain it, but let me just give you a brief stages of the cell cycle, okay? Now, the cell cycle or the cell division cycle, it is a series of events that take place in a cell leading to its division and duplication of the DNA. So then we have what we call DNA replication. That means the DNA will double to produce 2 daughter cells. It will double to produce 2 daughter cells. Now, the cell cycle consists of 4 distinct phases, like I said previously. We have the G1 phase, the S phase. The S phase is also known as the synthesis phase. We have the G2 phase, also known as the interphase. Then we have the M phase, known as the mitosis or the meiosis phase. Now, DNA replication occurs during the S phase. I repeat, DNA replication occurs during the S phase. During the S phase. The S phase. So over here, we are having what DNA replication or duplication, and it takes place during the S phase. The S phase. The S phase. All right. A 28-year-old female patient consulted a gynecologist about sterility. Examination revealed underdeveloped ovaries and uterus. This is a 28-year-old female. We have underdeveloped ovaries and uterus. Irregular menstrual cycle. Analysis of the sex chromatin revealed 2 BAHA bodies. Guys, do you remember when I talk about trisomy X? Yes, a girl must have only 1 BAHA body, but the person have 2 BAHA bodies. 2 BAHA bodies. This is what trisomy X. Trisomy X. So, what chromosome disease? I've already told you this is what trisomy X, also known as triple X syndrome. Triple X syndrome. Triple X syndrome. Again, Klinefelter is what? XXY, found in boys. Edward is what? Trisomy what? 18. Patao, trisomy 13. Tennis, XO. Thank you. A cell at the stage of mitosis. Anaphase was stimulated by colchicine that inhibit chromosome separation to the poles. What type of mutation would be caused? Now you know anaphase, chromosome must move to the opposite side. Again, cell division. This is where cell move to the opposite side. Cell begins to move. That's anaphase stage. Cell begin to move to the opposite side. So if there's a stoppage or there's a drug called the colchicine that inhibit the chromosome separation, what will it lead to? Definitely it will lead to what we call polyploidy. It will lead to what we call polyploidy. And polyploidy is the state of a cell or organism having more than 2 paired set of chromosomes. Having more than 2 paired sets of chromosomes. So if these sets are not moving, they will have more than what? Chromosome, more than 2 sets of what? Chromosome present. And that's what we call it what? Polyploidy. 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 So the answer is E. Is E. A patient in a transplantation center underwent heart transplantation. Again, if there will be a reaction, you are thinking of what? Type 4. Type 4. Because T cells will be involved. Now, the organ was taken from a donor who died in a road accident. Foreign hearts can be rejected as a result of transplantation immunity, which we said it's usually prevented. So how do we prevent this? You give what? Immunosuppressants. Give immunosuppressant to prevent reaction to take place. And immunosuppressant we give is what? Corticosteroids, to be specific, prednisolone. So here is what? Immunosuppressant or corticosteroids. It will suppress the immune system. Again, following an exposure of radiation. Sometime later, most of them were detected and destroyed by the following cell. Radiation affects which cells? Of course, the lymphocytes. So these are called what? The NK cells or the natural killer cells or the T lymphocyte killers. So going forward, T lymphocyte killers, the T cells. Radiation affect lymphocyte a lot. So the T lymphocyte killers will be destroyed. All right. Again, assuming you don't know this disease, look at what is going to happen here. Gynecologist was examining a patient with reveals symptoms of genital tract infection. Genital. A smear of the vagina contains piriform protozoa with spine flagella in their front. No, there's also an undulating membrane. There's also what undulating what membrane. What disease can be suspected? What disease can be suspected? Do you remember undulating membrane we mentioned earlier on? Yes, we are referring to what? Vaginalis or Trichomonas vaginalis. Trichomonas vaginalis. Trichomonas vaginalis. Trichomonas vaginalis. So basically, that is it. We've talked about this. We said it has the shape of a pear shaped with a central nucleus and 4 anterior what? Flagella. And it has an undulating membrane. So trichomonas, of course, vagina can also mean urogenital. So your answer is B, urogenital. Okay, look at this. A boy has blood group O and his sister blood group what? 4 or AB. What blood groups do their parents have? Blood group O and then blood group what? AB. So what kind of blood group are your parents having? Now look at this. This is having a blood group O, that means OO. And they've even shown you the genotype over there. Is what is OO. OO. And this one is what? AB. 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 So what it means is that their parents must have at least, now this chart OO, that means one O comes from the mother, one O comes from the father. True or false? Of course, true. So if it is true, that means that mother, which is M, and father, which is F, must have the O, and father must also have what? O. Mother must have O, father must have O to get what? OO in this chart. Yes. And again, for the sister to also have AB, that means mother must have either A or B, or father must have either A or B. In that case, we'll have what? A, B. So that means your blood group or the blood groups from your parents will be what? OA and then OB. OA and then OB. So let's look at our options. So where is OA? OA is the same as blood group II, right? And then OB is on blood group III. So III. So your answer is what? Is A. As simple as ABCD. Very interesting, isn't it? It is. For the purpose of myocardium infarction treatment, a patient was injected with embryonal stem cells derived from this. Look, there is a stem cell from this same patient by means of therapeutic cloning. That means that they are taking part of the person's body or part of the person's stem cells to the myocardium site or to the place of surgery. And that is called what? Autotransplantation. So your answer is what? Is B. Your answer is B. Now, in allo transplant, it is where the transplantation of the cells or tissues or organs to a recipient we transfer these things from a genetically non-identical donor of the same species. Genetically non-identical. So from someone, basically. The author I've already explained. Xenotransplantation is from other species. Let's say you take something from a pig to put inside a man. It's called xenotransplantation. So please take note of some of these things. All right. An alcoholic woman has born a girl with mental and physical developmental lack. Doctor diagnosed the girl of what? Fetal alcohol syndrome. Fetal alcohol syndrome. That means the mother was what? Alcoholic. And we said any drug that will lead to congenital defects, we call it what? We call it a teratogen. A teratogen. A teratogen. That is what? Genetic. Sorry, that is what? Congenital defect. Now, in DNA defect or chromosome defect, we are referring to what? A mutation. 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 But in physical defect or birth defect, we are referring to what? Teratogen. So this is what? Teratogenic. 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 So the answer is E. All right. Again, we have answered this question. Look, sex chromosome of a woman did not separate and move to the opposite pole during cell division. The ovum was impregnated with a normal spermatozoon. Which chromosomal disease can be found in this child? So now let's look at it. Look at Turner syndrome is what? XO. That means the X didn't come. The other X didn't come. Down syndrome is trisomy 21. Patao 13. Edward 18. So that means we're referring to what? Turner's disease or Turner syndrome. Turner syndrome. Turner syndrome. Turner syndrome. Because chromosome did not separate. So it didn't come down. All right. Hartnup disease is caused by a point mutation of only one gene, which result in the disturbance of tryptophan absorption in the bowel and its resorption in the renal tubules. It is also the reason for the disorder of both digestive and urination system. What genetic phenomenon is this? Guys, when one gene, influences two or more seemingly unrelated traits, that phenomenon is called pleiotropy. 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 So a pleiotropy occurs when one gene influences two or more seemingly unrelated phenotypic traits. Unrelated phenotypic traits. Thus, one gene defect contributes to multiple effects. One gene effect or defect contributes to multiple effects. To multiple effects. To multiple effects. Example is the Marfan syndrome or the Hartnup. This is a genetic disorder of the connective tissue, and this defect is causing all the above phenotypic disorders. It's causing digestion disorder, urination disorder, absorption of some vitamins or nutrients. They are all being affected. So this is what? Pleiotropy. Pleiotropy. No, this is not this is pleiotropy. This is E. Pleiotropy, not codominance. Not codominance. Codominance is like somebody having blood group AB, where the A and B, they are all co-dominating, or they are all there, functioning effectively. None is recessive to the other. All of them are dominant. Cytogenic examination of a patient with reproductive dysfunction revealed a normal karyotype, but most cells have karyotype of Klinefelter disease syndrome, where 47XXY. Such cell heterogeneity is called, so this is also called what? Mosaicism or mosaicism. 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 And this is simply having more than one type of cell in a person. Having more than one type of cell. Having more than one type of cell. More than one type of cell. Example is what we have in this person. XXY is having more than one type. More than one type. So chromosome is 47. But normal chromosome is what? 46. This person is having what? 47. 47, 47, 47. All right. So here we are having what? Mosaicism. 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 All right. As a result of a prophylactic medical examination, a 7-year-old boy was diagnosed with Leschnehan syndrome. I think we've done this before. Leschnehan syndrome. Only boys fall ill with it. Only boys. So let's see, if it's only boys, that means either the father must have it or something like that. The boy's parents are healthy. Oh, boy's parents are healthy. But look, his grandfather, by his mother's side, suffers from the disease. That means that the woman is actually having the disease. Is the woman having the disease? That means the woman is a carrier. The woman is a carrier. The woman is a carrier because the woman got it from the father. And the father gave her what? The X chromosome. So that means this is X chromosome related. And it is a recessive since the mother is healthy. That means the mother is a carrier. So that means this X chromosome is recessive, or this gene on the X chromosome is recessive. Is recessive. So therefore, our answer is what? It's a recessive sex-linked or recessive X-linked. 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 All right. A doctor revealed tissue injury on a patient's scalp with localized suppuration and diagnosed. I think I'll tell you about what? Myiasis. Myiasis. This infection is caused by, and your answer is what? Warfarin fly. Warfarin fly. And this myiasis is a parasitic infestation of the body of a live mammal by a fly larva called the maggot. And these maggots grow inside the host while feeding on its tissue. So again, this is typical for warfarin or no, Walfat fly. I think it's a German word. It's a German word. Word. So basically, your answer is what? Is E. Your answer is E.